the next module on parenteral dosage form. In the previous module, we have discussed about introduction of parenteral dosage form, pre-formulation of parenterals and formulation aspect. In this module, we will be discussing about different types of parenteral preparation. After learning this module, you will be able to understand different types of parenteral products, their basic properties like particle size, syringeability, injectability and globule size and different methods of preparation of parenteral products. We will be covering drug injection, that is injectable solution, emulsions, suspension and dry powder, characteristics and properties of different types of parenterals, method of preparation of injectable solution, suspension, emulsion and dry powders. Now, moving to the different types of parenteral preparation. There are small volume parenterals, large volume parenterals and modified release or sustained release injectables. Small volume parenterals, when the injectable volume is less than 100 ml and the solution, suspension and emulsion and dry powders, they are a small volume parenterals. Large volume parenteral dosage form, when the injectable volume is more than 100 ml. The example for that is irrigating solution, dialysis fluid, electrolyte solution, carbohydrates and proteins. Modified release or sustained release injectables, they are implants, liposomes, microspheres and they are delivered by the route other than IV generally. Method of preparation of solution type of parenteral dosage form. The steps involved in method of preparation are accurately weighing the quantity of active pharmaceutical ingredient, then addition of suitable excipient, adjustment of pH and tonicity, and it is very important to adjust pH and tonicity, and we have discussed in previous module about importance of pH and tonicity for parenteral dosage form. Then filtering, sterile filtration through 0.22 micrometer membrane filter is used for filtration, and then final sterilization by using suitable method, then packaging and labeling. These are the steps involved in preparation of solution type of parenteral dosage form. Sterile filtration is used for thermolabile substances. Thermolabile substances, those substances which are degraded when they are exposed to higher temperature, and for sterile filtration 0.22 micrometer membrane filter is used, and sometimes carbon and talc is also used as an filter aid, and if talc is used, it should be pre-treated with dilute acid to remove the impurities of surface alkali and metals. In case of proteins, to avoid the adsorption of protein onto the membrane surface, a specialized membrane such as hydrophilic polyvinylidene hydrofluoride and hydroxy modified hydrophilic polyamide membranes are used. For thermostable substances, these are sterilized by autoclaving, and the standard temperature of autoclaving and pressure is used. The standard temperature is 121 degree centigrade for 15 to 20 minutes at 15 psi pressure. Small volume parenterals, they are ampoules, solution or liquid fill concentrate, vials, plastic blow fill seal ampoules, pre-filled syringes and needle free injections. So, these are small volume parenterals. Suspensions, parenteral suspension is dispersion of solids into suitable vehicle. It can be of oil or water depending upon the solubility of active pharmaceutical ingredient. Suspensions are one of the most typical and difficult formulation with the preparation aspect, and it cannot be administered by IV route as it need to fulfill the requirement of syringeability and injectability. And syringeability and injectability, this is also we have discussed in our previous module, that syringeability is handling characteristics of a suspension and injectability is with reference to the force and pressure required during injection. So, the requirement of ideal parenteral suspensions are: the very first requirement, maintenance of particle size distribution, zeta potential. Zeta potential, it is important as it contributes in the stability of suspension, rheological properties. The stability of suspension increases on increasing the viscosity. So, by adding some amount of protective colloids, for example, sorbitol or any other suitable protective colloid can be used and the suspension can be stabilized. Surface activity, wetting and contact angle, these are important properties of suspension, and the solid content ranges from 0.5 to 5%, and it may go as high as up to 30% in some cases of antibiotic preparations. Redispersibility, this is also important characteristics of suspension, that we should avoid the formation of cake, and it can be prevented by the use of surface active agent. It reduces the interfacial tension between the particle and vehicle, and for example, polysorbate 80, lecithin, hydrocolloids like sodium CMC. So, these are surface active agents which are used in the preparation of suspension type of parenteral dosage form. Now, moving to methods of preparation. There are two basic methods used to prepare parenteral suspension. In the first method, for example, preparation of procaine penicillin injectable suspension, an aqueous vehicle containing soluble components such as sodium citrate, povidone or lecithin, it is filtered through 0.22 micrometer membrane filter, is heat sterilized and transferred to a pre-sterilized mixing filling tanks. Then the sterile antibiotic powder, which has previously been produced by freeze drying, sterile crystallization or spray drying, is aseptically added to the sterile solution while mixing. So, all the tests have been completed on the bulk formulation and then it is aseptically filled. So, this is first method for preparation of parenteral suspension. In the second method of preparation, for example, preparation of testosterone suspension, the vehicle is prepared and sterile filtered. The testosterone is dissolved separately in acetone and sterile filter. Acetone, it is used as a co-solvent. The testosterone acetone solution is aseptically added to the sterile vehicle and causing testosterone to crystallize. The resulting suspension is then diluted with sterile vehicle, mixed, and crystals are allowed to settle and the supernatant solution is siphoned off. This procedure is repeated several times until the complete acetone is removed. So, this is the second method for the preparation of suspension type of parenteral preparation. Now, there are few official preparation of parenteral suspension, for example, sterile ampicillin suspension USP and sterile tetanus toxoid adsorbed suspension and betamethasone acetate suspensions. So, these are few official preparation. Now, moving to the stability and evaluation of parenteral suspension. So, these are the parameters which are important to evaluate. These include injectability, freeze thaw cycles, resuspendability, sedimentation volume, crystal growth, particle size measurement, zeta potential and viscosity of suspension. So, these two properties, injectability and syringeability, they are closely related to the viscosity and particle size. So, we have to ensure that the entire suspension passes through 25 gauge needle syringe with internal diameter of 0.3 mm, and freeze thaw cycles, it determines the suspension ability to withstand with thermal shock, crystal regrowth. So, an appropriate number of freeze thaw cycles are decided. Resuspendability describes the ability of suspension to uniformly disperse with minimum shaking after it has stood for some time. So, qualitatively, light transmittance through upper solution in a cylinder after it has spun for 2 minutes at 75 rpm can be used to detect the dispersion properties of suspension. Sedimentation volume, it is defined as the ratio of final volume to the original volume of the suspension. The larger the sedimentation volume, better is the suspendability. Crystal growth in suspension is affected by the particle size distribution, change in pH and temperature. So, the crystal growth should be monitored by examining changes in particle size over time and comparing that with the initial particle size distribution. Particle size measurement, they are useful in that they allow aggregation or crystal growth and to be evaluated, and there are number of methods used for particle size analysis. So, microscopic determination is generally preferred over Anderson pipette or sub-sieve sizer, and turbidity methods is also used sometimes. Zeta potential determinations. Zeta potential is determined by zeta meter, which is a micro electrophoretic mobility apparatus used to characterize flocculated and dispersed suspension and to follow the change in physical stability with time. Next is viscosity. So, an ideal suspension should exhibit a high viscosity at low rate of shear. So, a suitable method can be used to determine the viscosity of suspension. Moving to emulsion type of parenteral preparation. Emulsions are dispersed system, it is dispersion of oil in water or water in oil. They are milky white in appearance and having globule size of 1 to 5 micrometer, and globules larger than 5 micrometer should be less than 0.05% of total volume. For example, propofol and oil soluble vitamins, they are example of emulsion type of parenteral preparations, and you can refer more example. Emulsions, the particle size should be within the permissible limit as to prevent the formulation of emboli in blood vessel and preventing the ranicity of oil phase. And the stability is important as sterilization should not affect the stability of the product. And the small amount of stabilizers like gelatin, dextran and microcrystalline cellulose, they are added to stabilize the emulsion. Now, the example of parenteral emulsion, lipid emulsion for parenterals, they are generally given to the patient to give the nutritional value. So, they are widely accepted for the patient who are unable to take food by themselves, and it consists of a suitable oil and emulsifier and a balanced blend of amino acid in a continuous phase of distilled water. The composition of emulsifier, it is important as it decides the overall stability of the emulsion. These emulsions are injected into the vein and it provides, as I mentioned earlier, a suitable mean of administering a nutritive fluid to the patients. So, the other example of emulsion type of parenterals are perfluorochemical emulsion. This type of emulsion utilizes perfluorocarbon oil and emulsifier Pluronic F68. It was used in the original formulation, hydroxyethyl starch and various salts in water. So, perfluoro oil is the oxygen carrier, that is the oil can dissolve the oxygen and carry it through the body. So, these emulsion have great deal of potential for the following reason. Number 1, non-availability of compatible blood, bloodless surgery for the treatment of various diseases. Liposomes are emulsified spherical vesicle composed of phospholipid bilayer with an aqueous inner phase, and the drug can be incorporated either in the lipid phase or in aqueous phase. So, liposomes are another example of parenteral emulsions. Now moving to dry powders. Dry powders, these are the preparation required to be reconstituted before administration. So these type of preparation are more suitable for those drugs which are too unstable either physically, chemically or in aqueous medium. The reconstituted product is usually aqueous solution and occasionally it may be an aqueous suspension, for example penicillin trihydrate. Dry powders for reconstitution as an injectable product may be produced by the following method: aseptic crystallization and dry powder filling, spray drying and freeze drying. So these are three methods which are used for the preparation of dry powders. Moving to aseptic crystallization. In aseptic crystallization, the drug is mixed with the solvent, an appropriate solvent is used, it is sterile filtered, then it is added to the non-solvent and the solution is kept for crystallization, and after crystallization the crystals are collected, and if it is required the blending and milling of crystals is done and then final packaging. So aseptic crystallization, this is one of the method for preparation of dry powders. Moving to the second method, spray drying. In spray drying, sterile filtered solution of drug is metered into the drying chambers where it passes through an atomizer that creates an aerosol of small droplets of liquid. The aerosol, it comes in contact with the steam of hot sterile gas, usually air is used. The solvent evaporates quickly, allowing drug to be collected as a powder in the form of uniform hollow spheres, and this powder is then filled into the vials. Now moving to freeze drying, a solution is filled into vials, a special slotted stopper is partially inserted into the neck of vial and trays of filled vials are transferred to the freeze dryer. There the solution is frozen by circulation of a fluid such as silicone oil at a temperature ranging between minus 35 degrees centigrade to minus 45 degrees centigrade through internal channel in the shelf assembly. When the product has solidified sufficiently, the pressure is reduced to a pressure less than the vapor pressure of ice at a temperature of the product, and heat is applied to the product by increasing the temperature of the circulating fluid. Under these conditions, the water is removed by sublimation of ice. In the whole process of freeze drying, the critical quality attributes should be taken care of. It includes the dissolution, appropriate residual moisture level and acceptable cake appearance. In addition, the process condition should be those conditions that minimizes the drying time without adversely affecting the product quality. So this is the third method for the preparation of dry powders. So far we have discussed the different types of parenteral preparation and their method of preparation. We have discussed solution, suspension, emulsion and dry powders. Now to ensure the product meets the requirement of quality, each manufacturing process should be validated to be sure that it is accomplishing what it is intended to do. So FDA provides certain guidelines for current good manufacturing practices for finished product. So pharmaceuticals under CFR, that is Code of Federal Regulation, section 211 and its subsections for detailed requirements. CFR 25 subsection of 211, it explains the professional qualification requirements based on education, experience and training for the job, and subsections 80, 84 and 86 it describes ingredients used in product compounding, meets the requirement of identity, quality and purity, and subsections 63, 65, 67, 68 and 182 it describes the validation requirement for processes and the equipment used. Next, CFR subsection 42, 46, 56 and 113, it explains the requirements for the product environment preventing any contamination, and subsection 160, 165 and 167, it explains about requirement for quality control procedure ensuring the finished product has the required potency, purity and quality until the expiration date is reached. Now, moving to the large volume parenterals. As I mentioned earlier, large volume parenterals are having the injectable volume more than 100 ml. These are electrolyte solution, for example 0.9% of sodium chloride isotonic solution, 0.45% or 3% sodium chloride solution, and the Ringer lactated solution, etc. Carbohydrate solution, for example dextrose 5% in water. Nutritional proteins 2.5% to 10% synthetic amino acid mixtures used on patients with starvation, renal hepatic failure or other conditions. So, these are large volume parenterals, and the another example of large volume parenterals are peritoneal dialysis fluid and irrigating solution. Now, moving to the modified or sustained release injectable preparations, they are meant for prolonged duration of action, reduction in dosing frequency and increased patient compliance. For example, polymeric implant, microspheres and liposomes, they are modified sustained release injectable preparations. So, dear learners, so far we have covered the different types of parenteral products and their characteristics, properties, and we have studied the different methods of preparation of different parenteral products like solution, suspension, emulsion and dry powders. And please go through from the suggested further readings and do take part in our discussion forum if you have any query regarding the content of this module. In the coming module, we will be covering plant layout and production facilities for parenterals, and thank you so much for paying attention.